Hi guys, so in this video we're going to go over the HL content of A1.2 nucleic acids. If you haven't already watched the previous episode where we go over SL content as well, because we're going to be building on what we said earlier on and introducing newer higher level content. So to start, let's go over what I said about the directionality of RNA and DNA in the previous video. We're going to go in a bit more depth now, but we're going to learn about something called 5' to 3' linkages. Let me just draw the nucleosomes here. I mean, nucleotides, sorry. So this is how that's a sugar phosphate backbone that I just drew. That's a phosphodiester bond. This is what two nucleotides of a DNA strand look like, for example. So if we look at the molecule, we know that the pentose sugar has 5 carbons. So I'm just going to label this here. It's a bit weird, the labeling. It starts from here. 1, 2, 3, 4, and there's a fifth one up here. So 1, 2, 3, 4, and there's a fifth one up here. This point here is the oxygen, so it's not counted as one of the pentose carbons, I guess. So as we can see, the nucleotides are connected from the sugar phosphate backbone by the 5' carbon to the 3' carbon. This 5' is what connects to the 3' and so on. So each one's added to the third one. And so both strands in a DNA do go to a 5' to 3' direction, but because it's anti-parallel, the other one is just flipped, so it does go 3' to 5'. However, both strands are synthesized in a 5' to 3' direction, as I discussed in the DNA replication video and in more videos to come. So this directionality is significant. So if we see this is 5 to 3, and the other one would be going 3 to 5. Um, yeah, so well, as I was saying, this directionality is significant for processes such as protein synthesis, replication, transcription, translation, and so on. And basically, in replication, in all of these processes, these strands are synthesized in the same 5' to 3' direction. Um, so yeah, and the way I remember this is if I think clock, you always say, oh, it's 5 to any time. You never say 3 to something. It's always 5 to 3, you know? So, if you like retime in that way, 5 to 3 makes the most sense. Again, this might not work for everyone, but yeah. Now we're gonna go on to something called purines and pyrimidines. So we know about the 4 bases, or well, technically 5. Like A binds to T or U, depending if it's RNA or DNA, and G or C binds with C because of these hydrogen bondings, but we can split these up or group them into something called, or into two chemical groups called purines and pyrimidines. One thing you have to know about these is that purines are larger and pyrimidines are smaller, and this size difference allows for a stability within the DNA double helix. So each purine binds with a pyrimidine, and a pyrimidine binds with a purine. You can never have a pyrimidine-pyrimidine binding or a purine-purine binding. So if we look at, if we look at a strand, the length, the distance between the two strands will always be the same because a purine binds the pyrimidine and a pyrimidine binds the purine. It's never both binding to each other. Does that make sense? So that consistent distance allows for that stability. Um, so purines are adenine and guanine. Pyrimidines are cytosine, thymine, or uracil. So because they bind with each other, like A with T and U and G with C, that consistent distance between the two bonds, the two strands are allowed. The way I remember this is purine is pure as gold. So A and G, pure pyrimidine. And then cut the pi. But obviously, I mean pi, like pyrimidine. So that's how I remember it. Again, this might not work for everyone, but it does work for me. Now, we're going to go into something called a nucleosome. And this is just a structure that helps condense DNA, because as I discussed again in the SL video, DNA can be very, very long because of variation. So it must be coiled and wrapped tightly in order to compress it into smaller components like chromosomes, for example. So nucleosomes are what coil DNA to prevent knotting and tangling. What I mean by this is that in processes such as mitosis, meiosis, when the DNA strands are pulled to opposite ends of the cell for replication, um, they, they're vulnerable to knotting and tangling, therefore breakages, if they're not compressed, because it can be very long, the bases are like jutting out or whatever. So, to form a nucleosome, these DNA molecules are wrapped around something called 8 histone protein core. And so histones are a type of protein, and they're 8 because there are four different types, but two of each. And then there's one additional histone called an H1, which is sort of like a linker that prevents the unwinding. So I'm going to just try to draw this out for you guys right now. So let's see, there's one of type, this type, other. The DNA will come around, wrap itself around these, and come out. And that condensation, like that long strand, can be wrapped around these 8 histones and wrap around it properly. But it condenses it, and then that H1 comes around and sort of links it up so it doesn't unwind. And like I said before, this is very useful for packaging during like division process, like cell division and all that. One more thing to keep note of is that this is only in eukaryote cells. And that's because plants, animals, eukaryotes have nuclei to store our DNA and whatnot. Bacteria or prokaryotes don't. They don't have a nuclei, so they don't require nucleosomes. Yeah. I'm gonna do something called the Hershey Chase experiment. It is a bit confusing, but bear with me. Basically, in the early 1900s, or mid 1900s rather, we weren't sure whether DNA or protein were the actual genetic material of a cell. And this confusion was largely because of viruses. So if we look at a virus, this is a drawing of one, but we know that the virus has a protein coat and it also has DNA or RNA. And we weren't sure whether if it was the protein coat or the DNA or RNA that they inject into a bacteria cell to infect them. So Alfred Hershey and Martha Chase in 1952 conducted some experiments which led us to prove that DNA was genetic material and not the protein or the protein coat. So what they did was they got two viruses. Well, they look like this, called T2 bacteriophages, and they basically cultured them in two different isotopic mediums. So one was cultured in sulfur and the other in phosphorus. Radioactive phosphorus, radioactive sulfur. And that's because phosphorus DNA has phosphate backbone, sugar phosphate backbone, and phosphate group. So it allowed phosphorus to be a marker for the DNA, and then sulfur was for protein. Let me just roll that down. For DNA, protein. Now, what basically happened was the viruses were cultured, then they were allowed to infect E. coli, which is a type of bacteria, and then they underwent something called centrifugation. So they were centrifuged. And what this process is basically these little tubes are filled with a solution, and they're put in this machine that spins them around um to separate solutions of different densities into different densities. Like they make a clear distinction, and that's through the use of centrifugal force. And basically, what happens is the result of that is that there's super liquid that's less dense called the supernatant, and a little pellet forms at the bottom. This is solid. And this one had the bacteria after centrifugation because the bacteria was more dense than anything else above. So Hershey and Chase tested this bacteria and found it to have strains of the radioactive phosphorus. And that's how we know that it was DNA that's the genetic material and not the sulfur, because whatever we find in the bacteria should be the genetic material, thanks to the theory that we know that viruses inject their genetic material into bacteria or whatever they're infecting to infect. And yeah, that's pretty much it. Now, the next is everything almost written out for you, but we're going to learn about something called Chargaff's rule. Again, this is a bit confusing, but it's yeah. Originally, we had something called the tetranucleotide hypothesis, and in that, they stated that all the bases were of the same proportion. So, if C was 25% of like CATG, were all 25% of the like genetic material, whatever was found in the organism. And technically, this could make sense because of complementary base pairs. Um, but Targoff, was like, okay, but if that was the case, there wouldn't be enough variation because then everyone would have the same portion of base sequences. And as I mentioned earlier in the SL video, the DNA sequence is what gives us that actual genetic material that codes for these genes and proteins and whatnot. So, Erwin Chargaff, what he did was he analyzed some data samples over a range of species to find the nucleotide composition, and he provided this hypothesis. DNA contains the same number of adenine as does thymine and the same number of cytosine as does guanine. And again, this may sound really similar, but there are slight nuances. And he uses the theory of complementary base theory to explain it. So we know that A binds with T and C binds with G. So how many ever A's there must be the same T's. And however many C's there must be the same G's. However, there's no connection between the A and the C or the T and the G because they don't really matter. They aren't like complementary base pairing with each other. So that plays no effect. So if we take this on a more math route, let's say 20% of the genetic data or genetic genome is A. That must mean 20% is T because of complementary base pairing. And this equals 40%. So 60% must equal C and G, and therefore 30% is C, 30% is G, because of, again, complementary base pairing. But notice how these proportions are not exactly the same. They're not all 25%. They have their own proportions in relation to one another. And I know it's a bit confusing, but these subtle nuances is what allows DNA to have variation and allows for variation within our populations. Yeah, hope this video helped and see you next time. Thank you.